On behalf of the Patient Safety Authority, I would like to welcome you to this webinar titled Antibiotic Stewardship Webinar Series, Mechanisms of Antimicrobial Resistance. My name is Joanne Atkins, and I will be your moderator for this program. All materials are provided for your educational use. Now, I would like to introduce our speaker for the webinar. Amanda Bennett is an infection preventionist for the Patient Safety Authority. In this role, she serves as a resource for Pennsylvania facilities through education and the development and monitoring of infection prevention and reduction initiatives. Amanda has an extensive background in infection prevention, laboratory testing, analysis and interpretation of diagnostic results, education, and regulatory standards. Prior to joining the Patient Safety Authority, Amanda worked in acute care settings as a molecular pathology medical laboratory scientist, point of care coordinator, and infection preventionist. Amanda, I will now turn the program over to you.
Thank you, Joanne. I'd like to welcome everyone and thank you for your time today. We will be talking about mechanisms of antimicrobial resistance, and this is our fourth webinar in the Antibiotic Stewardship Series. The objectives for today's webinar are to discuss the misuse and overuse of antimicrobials and the resulting patient safety issues, identify the major mechanisms of antimicrobial resistance, and explain how resistance develops in microorganisms. To really understand how extensive the threat of antimicrobial resistance is, we need to look at the numbers. The CDC considers antimicrobial resistance an urgent global public health threat. According to a 2019 report released by The Lancet, antimicrobial resistance has killed at least 1.27 million individuals worldwide. And from the 2019 CDC report on antibiotic resistance threats, we also know that more than 2.8 million antimicrobial resistance infections occur each year in the United States alone, resulting in over 35,000 deaths. The CDC's 2019 Antibiotic Resistance Threats Report showed that dedicated prevention and infection control efforts in the United States reduced deaths from antimicrobial resistant infections by 18% overall and by nearly 30% in hospitals from 2012 to 2017. However, the CDC released a 2022 special report which highlighted the impact of COVID-19 on antimicrobial resistance in the United States. It found that much of that progress was unfortunately lost. This was in large part due to the effects of the pandemic. The use of antibiotics really varied during the pandemic based on location. In hospitals from March of 2020 to October of 2020, almost 80% of patients which were hospitalized with COVID-19 received an antibiotic. Conversely, in outpatient settings, antibiotic use significantly dropped in 2020 compared to 2019 due to less use of outpatient health care and less spread of other respiratory illnesses that often led to antibiotic prescribing. This did rebound, however, in 2021. In nursing homes, antibiotic use spiked along with COVID-19 surges, but fortunately has overall remained lower than before the pandemic. We have reviewed this previously during this webinar series, but to quickly review, how exactly does antimicrobial resistance happen? To start, we have a group of bacteria, some of which are drug resistant, but most of which are not. An antibiotic is prescribed to treat the bacteria causing this particular illness or infection, but this also kills the good bacteria which protect our body and keep down the growth of harmful bacteria. Because of this, drug-resistant bacteria are able to grow and take over. To further complicate the situation, some bacteria are able to give their drug resistance to other bacteria, which we will talk about more in later slides. This slide shows 5 different types of bacterial resistance mechanisms, which are altered target sites, drug inactivation, bypass, decreased permeability or efflux, and restricting access. We will cover each of these in more detail in the coming slides. First up is an altered target site. Normally, antimicrobials single out and destroy specific targets on an organism. But with this mechanism, organisms can change the target, so the drug can no longer fit and do its job. Examples of this include MRSA, in which a penicillin-binding protein, or PBP, is altered to PBP2A. In E. coli, the MCR1 gene adds a compound to the cell wall, so colistin cannot latch. And in Aspergillus fumigatus, the CYP1A gene is changed, so triazoles can't bind to the protein. Next is drug inactivation. Just like it sounds, this is when the organism is able to produce an enzyme which destroys or inactivates the antimicrobial. We see an example of this in Klebsiella pneumoniae, which can produce carbapenemases, or beta-lactamase enzymes that break down carbapenem drugs and most other beta-lactam drugs. We also have bypass, which is the ability to develop an alternate metabolic pathway to bypass the pathway that the antimicrobial targets. An example of this is seen with Staphylococcus aureus. Some isolates of this organism can bypass the effects of trimethoprim or sulfamethoxazole. Organisms can also use decreased permeability or efflux. In this mechanism, the bacteria are able to get rid of the antimicrobial through pumps in its cell wall. This removes the drug from the cell once it enters. Examples of this are seen in Pseudomonas aeruginosa, which can produce pumps to get rid of fluoroquinolones, beta-lactams, chloramphenicol, and trimethoprim. In addition, some species of Candida can produce pumps to get rid of azoles, such as fluconazole. In the last mechanism we are going to discuss, the bacteria has an ability to restrict access. Organisms have an outer membrane in place to protect it from the environment. They are able to use this membrane to change entryways or limit the number of entryways in the membrane. An example of this is the ability of some gram-negative bacteria using its membrane to selectively keep antibiotics from entering. When we talk about the development of antimicrobial resistance, we have two potential ways that organisms can develop antimicrobial resistance: point mutations in existing genes or the acquisition of new genes. Point mutations are random errors that occur during DNA replication. When this happens, there is a substitution of one base pair for another base pair. With this, there is the potential that a substitution of one amino acid for another amino acid in a protein structure or enzyme can occur. Point mutations infrequently occur at the exact correct location on the bacterial genome required to cause resistance. If a point mutation is responsible for antimicrobial resistance, it usually creates a structural change in a drug receptor or target site. The second and more common type of antimicrobial resistance occurs from newly acquired genes. Newly acquired genes are more often the cause because most forms of antimicrobial resistance requires complex structural or enzymatic changes. We will talk more about the acquisition of new genes in the next slides. Resistance in organisms can be inherited or passed down generation to generation, or they can be passed directly from one organism to another through mobile genetic elements. Mobile genetic elements are also known as jumping genes, which allow for transmission of antimicrobial resistance between strains of organisms. The first example are plasmids, which are circles of extrachromosomal DNA that can self-replicate. Transposons are segments of DNA which can insert themselves into the chromosome and can then be transmitted between cells by way of plasmids or viruses. Then we have phages or bacteriophages, which are viruses that infect bacteria. The concerning risk with antimicrobial resistance transmitted by these mobile genetic elements is that they can potentially be spread to different strains or between different species of microorganisms. Some resistance genes that are transmitted in this way can be stably inserted into an organism's chromosome and can then be passed down by cell division. So how do these mobile genetic elements work? The first is transduction, which is associated with phages. Phages bind to specific targets on bacterial cell surfaces and are then able to inject their genomes into the bacteria and replicate. Antibiotic resistance can also be transmitted among bacteria through conjugation, which is a horizontal gene transfer mechanism. Conjugation occurs when there is a physical contact between two bacteria. Transformation occurs when a bacterial cell dies and bursts. With this, the intracellular contents are released and can be taken up by a living bacteria. If these fragments of DNA are incorporated into the chromosome of the living bacteria, it can acquire new characteristics, including antimicrobial resistance. Before we go into antimicrobial susceptibility, I want to cover some basic terminology. First, we're going to look at the way you'll see results. So the first is a susceptible organism. So this is that the drug is likely to be effective for the treatment of the infection using a standard dosage. Intermediate means that the drug is likely to be effective, but only at body sites where it's physiologically concentrated or at other body sites if we use a higher than usual dosing regimen. Resistant means that the drug is unlikely to be effective for the treatment of the infection unless it's used at toxic dosages. And then we have the minimum inhibitory concentration, or the MIC. This is the lowest concentration of drug that can still be used to inhibit microbial growth. Next, I'd like to go over some of the major ways in which we test for antimicrobial susceptibility. I think it's important to understand how the testing is performed so we can better understand and interpret the results. The three testing methods we will cover are microbe titer broth dilution, Kirby-Bauer or Auger disk diffusion, and the E-test, or the antimicrobial gradient diffusion method. While I do have text on this slide and future slides, I'm going to advance to the next slide while I talk about this method so you can see what I'm talking about as I explain it. Microtiter broth dilutions are done with liquid growth media. Identical amounts of bacteria are introduced into the wells containing liquid media with progressively lower concentrations of the antimicrobial agent. Results are read after incubation, and the MIC of the antibiotic is determined by looking at the microbial growth in the wells. The MIC will be between the last well, where we see no bacterial growth, and the next lower dose, which allowed bacterial growth. This type of test is also available commercially. So when we're looking at this photo, you can see that the wells with bacterial growth look hazy. Each well has the same amount of bacteria, and starting from the left has decreasing amounts of the antimicrobial agent added. Each row has a positive and a negative control in the last two columns. The far right is all of the positive control so that we can see the organism was able to grow, and then the next one over is the negative control. The serial dilution can be seen in columns 1 through 10. In row A at the very top, the MIC would be between columns 1 and 2. In row B, you see an organism that was able to grow in all wells, indicating that this antimicrobial agent was not able to inhibit growth at any concentration. And in row C, we see the MIC between columns 3 and 4. If you look closely, you can see that the wells get increasingly hazy as the concentration of the antimicrobial agent decreases. Next is Kirby-Bauer tests. These are done by disk diffusion antibiotic sensitivity testing on solid growth media. With this method, a known quantity of a pure culture bacteria is grown on an agar plate with antibiotic-containing discs. Results are then read after incubation by measuring the zone of inhibition. The zone of inhibition is the area on the media where bacteria are unable to grow due to the presence of an antibiotic that impedes their growth. The larger the zone of inhibition around the disc, the more sensitive the bacteria is to that antibiotic. You can see here an example of a technologist measuring the zone of inhibition around an antibiotic-containing disc. Kirby-Bauer results are reported based on the size of the zone of inhibition. There are standard size zones established for each organism and antibiotic combination. If the observed zone of inhibition is greater than or equal to the size of the standard zone, the microorganism is considered to be sensitive to the antibiotic. On the other hand, if the observed zone of inhibition is smaller than the standard size, the microorganism is considered to be resistant. The E-test is an antimicrobial gradient method performed on solid growth media. With this method, a known quantity of pure culture of bacteria is grown on an agar plate with a plastic strip that's impregnated with a concentration gradient of a specific antibiotic. After incubation, the results are read looking at the point of intersection of the growth ellipse and the inhibition zone on the strip. As you can see in this photo, we have the highest concentration of the antimicrobial agent at the top, and it decreases the further we go down on the strip. Here, the growth ellipse can be seen as the clear area around the plastic strip. This area shows where the bacteria were unable to grow due to the antimicrobial impregnated on the strip. As the concentration of antimicrobial lessens, the growth of the bacteria is able to come closer and closer to the strip until it is no longer inhibited and can grow right up to the strip. This is a sample of a susceptibility report. Each antibiotic has a reported MIC and an interpretation. The MIC indicates at what dilution the culture was sensitive to that particular antibiotic. This is then compared to the known breakpoint, or the dilution where the bacteria begins to show resistance for that antibiotic. This will determine if the organism is sensitive, intermediate, or resistant to the antibiotic. For example, the MIC for aztreonam is 8. The lab checks this against their known breakpoint and determines that this indicates the culture is sensitive to aztreonam. When selecting an antibiotic, a few factors need to be considered. This can include, but not be limited to, the body site, the health of the resident, any allergies or side effects of the drug, and the route of administration. So we've talked about how resistance happens, and part of this infographic from the CDC really reinforces why antimicrobial stewardship is so important. You can see how quickly resistance was able to develop from the time an antibiotic was first approved or released. Here we see penicillin was discovered over 90 years ago, and since that time, development of new antibiotics has slowed, but the development of resistant mechanisms has not. As you can see here, some organisms were able to very quickly develop resistance to certain antibiotics. Penicillin was first released in 1941, and the next year, by 1942, we saw penicillin-resistant Staph aureus. In addition, methicillin was released in 1960. That very same year, methicillin-resistant Staph aureus was identified. This is a continuation of the same infographic from the CDC, which shows more quick successions from the time an antibiotic was released to the time a resistant organism was identified. I would like to end this presentation on a good note. We all know we need more research and development aimed at producing more antibiotics, and in April of this year, the FDA Antimicrobial Drugs Advisory Committee unanimously voted 12-0 in favor of recommending the approval of a new drug called sulbactam durlobactam. This is an intravenous drug, which is the first pathogen-targeted therapy for Acinetobacter. Acinetobacter is largely considered intrinsically resistant, and the CDC's 2019 Antibiotic Resistant Threats Report that we mentioned at the beginning of this presentation has it listed as one of the 6 urgent threats. The indication for this new drug is hospital-acquired bacterial pneumonia and ventilator-associated bacterial pneumonia. Next up for this drug is the Prescription Drug User Fee Act, which is scheduled on May 29th, 2023. So you want to look for more updates on this towards the end of the month. Joanne, I will now turn the program over to you.
Thanks, Amanda. Excellent presentation. Now we would like to begin our question and answer period. Amanda, we did have one question come in. Do we need to be checking all antibiotics prescribed in the facility?
So this question I can kind of see two ways of asking it. So checking all antimicrobials from a surveillance standpoint, yeah, we should check any new antimicrobial starts to see if we can identify this as an infection that needs to be reported. If we're talking about it as part of our ASP program, we should really have this sort of established through leadership on the clinician side so that there is a common thread to what we're prescribing for certain infections, depending on, the body site or the susceptibility that we're seeing based on the biogram for the facility.
Thank you. Another question just came in. For microbiology testing, is one method preferred over the other according to the CLSI standards?
So the lab really has determinations on what they're going to use. There isn't necessarily one way that's better than another. Some organisms grow better on solid media, some organisms grow better in liquid media, but it's not a one way is better than another. Most laboratories have determined what they're going to do based on the specimens that they get, the equipment that they have in their laboratories.
Okay. Another question just came in from the same person. Recent update with MIC breakpoints for AST does not correlate with FDA breakpoints in package inserts. What are your recommendations for standardization?
I would say that is a conversation that you need to have with your laboratory personnel. They're really going to be the ones that determine what they're going to use. Honestly, I didn't know about this inconsistency, so just on the fly, I'm not really sure. But I would say that would be a conversation to have with your lab. They're going to look at their standards and what's expected within their community, what's being accepted. They're most likely going to have to go with what is seen in the testing method pamphlet and what is known with that instrumentation that they're using.
Thanks, Amanda. We have another question that came in. How do you select which antibiotic to use when there are multiple that are susceptible?
So this is really going to depend on the specific organism, the body site that it's coming from. Are we looking at a blood or a wound? It's also going to depend on, the residents. Certain residents may have allergies, or if they are a more high-risk resident. And then also the way that we can give the antibiotic. If we can give it intravenously or orally, depending on if the resident is long-term care or if we're in acute care, if they're looking at getting discharged. So there's a lot of factors that really go into that decision. It's not necessarily a one-size-fits-all that you can say for this organism, you should always use this antibiotic.
Thanks, Amanda. From infection prevention standpoint, I'll just read what the question says. From infection prevention standpoint to collect MDRO, how are you working with the pharmacy team beyond simply antimicrobial selection?
I guess it's more, how do you work with your pharmacist? So your pharmacist is going to be able to, really make recommendations on what they're seeing with the type of organisms, what they know will work better based on the body site. They are also going to be able to make recommendations based on what they're seeing, even maybe with other organisms that they're seeing. I'm sorry, I'm trying to answer the question, but I don't. Can whoever asked it clarify it maybe? So the pharmacy is going to be able to, again, the pharmacy is going to be able to tell you based on what they know about the mechanisms of how the antimicrobial works, which one might be better, depending on, like I said, the site of the organism, the resistance that we're seeing in the facility. They're also going to have a lot of recommendations to clinicians based on what they should prescribe for what we're seeing based on the antibiogram in the facility.
Thanks, Amanda. I want to thank everyone for joining us today. If you experienced any issues accessing the evaluation or the Certificate of Continuing Education, please feel free to direct any inquiries to Shelley Mixel at shmixell@pa.gov. This concludes our webinar.